Güney xan Amerikada postdoc edib və bu Texas Universitetində stem sellərdə, neural stem sellərdə exosomların, yəni ki, hüceyrə xaricinə ifraz olunan vesicle'ların və bunların fərqli patologiyalarda necə təsir göstərdiyini bizə öyrənib və öz biliklərini bugün bizimlə paylaşacaq. Biz çox şadıq ki, bizimləsiz və səbirsizlə sizin çıxışınızı gözləyirik.
Good morning, dear organizers, dear guests of today's conference. First of all, I would like to say thanks for the neuroscience organization for invitation and for kind introduction. On next few minutes, I'm going to talk about the therapeutic efficiency of the exosomes, stem cell-derived exosomes on neurological diseases. As we know, neurological diseases, they represent the prevailing pathological conditions among the most severe diseases worldwide, affecting approximately 10 million people. These diseases include neuromuscular diseases, dementia-related Parkinson's disease, Alzheimer's diseases, neuromuscular diseases, then brain tumors, etc. And the pathogenesis of most of these diseases is closely associated with the neuroinflammation and aggregation of the proteins in the brain tissue. The development of effective therapeutic treatment strategies for neurological diseases remains challenging. First of all, because the intricacy of the brain diseases, and second one, because of the blood-brain barrier, which restricts entrance of more than 95% of the macromolecules which were considered for the drug development. The blood-brain barrier permits entrance only for the lipid-soluble substances with the size 400 Dalton, and therefore also the short-term effects of symptomatic treatment for neurodegenerative diseases and those dependent side effects of these pharmacological agents force the medical community to seek more effective treatment strategies for this world health problem, and also to find more effective drug delivery systems to treat these neurological problems. And in this principle, the stem cell derived, the stem cell therapy, you know, that it becomes more effective. It attracted tremendous attention because it permitted to treat previously uncurable diseases. And on this context, it was found that their therapeutic effects is mostly related with the exosomes, with the different paracrine-like substances like cytokines, chemokines, and especially extracellular vesicles that they produce. The extracellular vesicles, they encompass a wide range of the particles with the sizes ranging from nanometer size till the micrometer sizes. But in this case, the exosomes, which are most commonly used as therapeutic agents, their size approximately from 30 to 150 nanometers. And these are all the paracrine effects that you see on the screen, which produced by mesenchymal stem cells or the neural stem cells, as you see here. They produce these substances, and for especially that's about the exosomes, which the size of from 30 to 100 nanometers, and they produce as a result of the fusion of microvesicular bodies with the cell membrane of the cell, and they release outside. So they will represent all the effects of the parental cell which produced them. But we have to underline that they will not exhibit the disadvantages of the cells which produce them. That's, you see here how they can be produced by how we can isolate the exosomes. It's by ultracentrifugation, the filtration, ultrafiltration, then size exclusion chromatography, ion exchange chromatography, and etc. These are the methods which are used to isolate the exosomes. And their content is very important to know that, as we see, they are complex protein systems that contain external proteins. These are tetraspanines, then alleles, and different antigen-presenting complexes. Also, they contain internal proteins, which are cytokines, chemokines, growth factors, then microRNAs, non-coding microRNAs, different types, which were characterized extensively for mesenchymal stem cells and for neural stem cells also. Then they contain metabolites, proteins, and all of them, they exert therapeutic effect on our cells. We see that how they affect the cells by, that is the cellular, their result is their therapeutic effect is achieved by different cellular and tissue reactions, which include, as we see, tissue regeneration, anti-inflammation, then the anti-cell death, anti-apoptotic, their effect. Also immunomodulation, antioxidative stress, and all of them, they are reached by the different signaling pathways. So as we see here, the advantages and disadvantages of the cell therapy, also the stem cell-derived exosome therapy. The advantage of the exosome therapy include their small size. It means that they can traverse the blood-brain barrier. They pass through it. Their membrane composition, their bilayered structure, their biocompatibility permits them to traverse these barriers and to incorporate into the cells. And also they can serve as cargoes for the therapeutic drugs. They can be carriers for the therapeutic agents that we can incorporate into these vesicles to target our specific tissues and cells. So then they are minimal risk for tumorigenicity, unlike stem cell therapy. Their immunological advantage that they permit them to escape the drug clearance issues. Also, they do not elicit immune response in the injected organs and organisms. Also, they do not cause vascular obstruction during systemic administration. So these are the advantages of the exosome therapy. And in our case, in our research that I'm going to present to you today, we used intranasal administration of the stem cell derived exosomes because there, as research showed, recent reports showed that intranasal administration of exosomes permit us to not only bypass the blood-brain barrier, but it is a very good route for entrance of the many substances into the brain tissue, as we see here, via olfactory nerve routes. They incorporate, and in our research we showed that they readily incorporate into neurons and microglial cells. So the aim of our investigation was to study the efficiency of intranasally injected neural stem cell-derived exosomes on the neuroinflammation. As we know, the neuroinflammation is the base for pathogenesis. It is the reason for development of many neuropsychiatric and neurodegenerative disorders like Alzheimer's disease and etc. That's why we tried to check, to examine the efficiency of the exosomes on this neuroinflammatory model. What we have done, we grew the iPSC cells. These are induced pluripotential stem cells, and we induced them to differentiate into neural stem cells. Then when they reached 90% confluency, we took supernatant culture medium and isolated from there via using ultracentrifugation, then ion exchange chromatography, and then size exclusion chromatography to isolate the exosomes. And then we characterize them by Western blot analysis, then by the electron microscopy. As we see here, we show the electron micrograph representation of the exosomes, which display the double bilayered membrane, the size of them. The nanocyte analysis showed their size and the numbers in our concentrate. Then the Western blot analysis showed their markers the tetraspanins just about I mentioned. So that was the characterization of the exosomes. And then we did our research. That is a research design, which experimental design of our research. What we have done, we took the male mice and 8 weeks old. They were subdivided into 3 groups. The first group was the control naive group. The second group, the second and third group, they received 7 days injections of LPS, lipopolysaccharide that we see. That's lipopolysaccharide, that outer cell wall of the gram-negative bacteria. And we injected it into the, we have done intraperitoneal injections of this LPS. During 7 consecutive days. Then on next week, we have done intranasal injections of exosomes for the third group. So the second group of animals, they received just vehicle, LPS and vehicle. The third group received intranasal injections, also intranasal injections of the exosomes besides LPS. Then on the third week, we have injected bromdeoxyuridine, bromodeoxyuridine, that is BRDU, which is the gold standard to check neurogenesis. And then we have done different behavioral tests to check cognitive and memory functions. And finally, on 10th week, we have done, we sacrificed animals and we took brain tissues to study them in histology and their histology and also the biochemistry. So what we have seen, I will escape these slides, and here we see the results of behavioral tests. So I will just, because of the limited time, I will just inform you that we saw the impairments of associative cognitive functions, associative memory functions, and cognitive functions. Also a little moderate impairment of the social interaction in the animal's behavior. So the social interaction behavior also was moderately reduced in LPS vehicle-treated animals. And then we have done the histological examination, immunohistochemistry, and immunofluorescence analysis of the brain tissues taken from those animals. And here we see the microglial cells. We know that microglia, that is innate immune response cells of the brain tissue, the main macrophages of the brain tissue. And we see the IBA positive cells. This is the marker for the microglia. And we have seen the increase of the number of microglial cells in the LPS vehicle-treated group. But the numbers were approximated the naive levels on the animals which received exosome therapy. Then we wanted to know if these cells, the microglial cells, are activated cells. For that reason, we have done double immunofluorescence staining, which we used. In this case, we used IBA1 together with CD68 staining. And we found that the double positive cells in LPS-treated, vehicle-treated group, that was increased in number. That shows that these are the activated microglial cells which increased in vehicle-treated group. But these numbers became, they normalized in EV-treated group, as we see in graphs shown here. That was done mainly in hippocampus of the animals. Each 15th section among the serial coronal sections through the brain tissue were examined for in our studies. Then we checked the inflammasome expression in those microglial cells. As we know, the inflammasomes, these are the main the markers. These are complexes, protein complexes, which assemble during the inflammatory reactions. So what we see, NLRP3 is then apoptosis-associated like protein, and they were also the triple staining we used for evaluation of the inflammasomes, and then we saw the increased amount of inflammasomes which which were expressed in microglial cells, also the amount per volume of the brain tissue. Then the biochemistry analysis also showed the increased amount of cytokines that are mediators of inflammasome activation, and the end products, which is interleukin-1, interleukin-18, they all were highly expressed in the brain tissue, which we examined for biochemistry. We used the sonication of the homogenates of the brain tissue of hippocampus, and we analyzed it for the expression of different pro-inflammatory cytokines. And we saw the increased amount of the cytokines in the brain tissue and in LPS vehicle-treated animals. In some regions, it was in EV-treated animals also they were high, but they mainly approximated the naive control levels. Then we examined the GFAP positive astrocytes in the brain tissue, and we found that the fractions, the area fractions of GFAP plus cells also have been increased in the hippocampus CA1, CA3, DG, dentate gyrus regions of the LPS-treated, vehicle-treated animals. And the numbers also have been increased in exosomes-treated animals, but they were less than in the vehicle-treated animals. So then we checked the neurogenesis. We checked the neurogenesis, and I will say shortly that BRDU expressing cells, neural, as you see, BRDU expressing cells in DG, that is the region of the brain where the new neurons are formed, the neural stem cells are located. And to check the extent of neurogenesis in hippocampus, we saw that amount of BRDU plus cells in the vehicle-treated LPS-injected animals, they were reduced. It means that neuroinflammation leads to, by increasing amount of the pro-inflammatory cytokines, it leads to decreased neurogenesis. It has negative impact on the neurogenesis that was also normalized by EV treatment. We saw it in BRDU new and plus cells, also in double cortin expressing cells, which are newly formed neurons. Finally, we have done Western blot analysis for synaptophysin and PST. These are the postsynaptic density protein, which are markers for the synaptic plasticity for the synapses. We saw that decreased amount of synaptophysin and PST expression in the brain, in the hippocampus of both groups of the LPS-treated or vehicle-treated, also of the EV-treated groups. So it means that EV treatment had moderate effect on synaptic plasticity, despite of its advantage for elimination of the neuroinflammation. So finally, what we saw, the intranasal administrations of iPSC, NSC neural stem cells derived exosomes in conditions causing the neuroinflammation is able to reduce neuroinflammation, to reduce amount of, it can moderate, it can modulate microglial activation and prevent neurogenesis decline in hippocampus. And it was efficient for maintaining the proficiency of many cognitive functions. So the exosome therapy is valuable. It has value in neurodegenerative diseases and in neurological diseases which are associated with a neuroinflammation, because it can reduce neuroinflammation and by that way it can preserve cognitive functions. So that is, as we see, the exosomes, they are sophisticated and very convincing agents for neurological problems. and which opens the door for paradigm shift in the contemporary medicine. And however, if despite the exosomes have many positive effects, have been confirmed as therapeutic agents, there are different barriers that should be overcome before their massive, widespread implementation. And these are the barriers that should be overcome. As we see here, we have to find the ways for large-scale production of exosomes. Second one, we have to find the high-throughput cellular sources for their isolation. There is needed implementation of reproducible and scalable protocols for their isolation and the production. Then we have to study kinetics of exosome therapy, exosome uptake by the target cells. Then we have to investigate, we have to research their biodistribution, their dosage, their cargo loading capacity, and the half-life of the cargo. Then we have to investigate the incorporation of the bioengineered ligands into the membrane that will increase their efficiency, their uptake by the specific target cells. That's all that I tried to say in this limited time. Thank you for your attention. Okay, any questions?
Thank you so much for your presentation. So, of course, we'll have questions. I already have some questions, but I guess I'll wait if... Yes. Okay, please.
Mm-hmm.
Thanks for your talk. Yes, I cannot agree more on the potential of exosomes for therapeutics. I think it's a nice path to explore. I think that another advantage of exosomes compared to traditional therapies is the ability to penetrate inside the neuron. So, for the third possibility, you deliver intranasal in order to achieve a widespread brain distribution. This is, of course, the best choice for disorders with a disseminated pathology, such as, let's say, Alzheimer's, frontotemporal dementia, and so on, where the pathology is spreading all over the brain. However, I was wondering if there is any chance to drive expression of exosome into a specific cell population. For instance, in biofilament Parkinson's disease, where the pathology is concentrated in the substantial library, I was wondering if there is any possibility to drive or to direct expression of exosome only to the familiar neurons instead of to the brain overall.
Targeted, you mean? Yeah. Thank you for the question. Yeah, we know that exosomes, they incorporate in different cell types, and as I understood, the question is how we can target specific cells by exosomes. As we all see in the literature, that there are different research is being done to specify, to target to develop the exosomes which can be incorporated directly into the specific tissue, into the specific cell types. And different cargoes are being developed to incorporate into the exosomes and then target the exosomes into the specific cell types. We just did, I skipped that slides when we incorporated exosomes into the intranasally, and 6 hours after the injection of exosomes, we cut the brain tissues, and after the 6 hours, we saw that we labeled the exosomes by PKH26, which in fluorochrome stain. Then we incorporated them into the brain, and we saw the expression of these exosomes inside of neurons, then microglial cells, then astrocytes. I did not stop there because of the limited time. We will hope that in future we will be able to produce such kind of exosomes that will be directly incorporated into the desired tissues. Thank you.
Do we have more questions? Otherwise, I'll spill the spot and maybe ask a quick question. You mentioned, I think the problem is not the problem, but the issue is the exosome delivery is a complex delivery, right? There are a lot of constituents. Can we fractionate, or has it been fractionated and delivered component by component to really understand what is the key things we are delivering with the exosome? Because I think at this point we're delivering myriad of things, proteins, lipids. So can we really put our finger on what is the key component?
Thank you for the question. Yes, we used in our study the neural stem cell derived exosomes, and they have been, in 2020, they have been extensively investigated, the content, the microRNA content, and the protein content. And it was found that these microRNAs and proteins which are inside of those particular exosomes, they have lots of functions, and there is a publication on that issue. The content of those exosomes that we use, there are lots of the anti-inflammatory substances, then pro-neurogenic substances, and other constituents. So the question is actually for bioengineering of the exosomes. That's also the open question nowadays. It's openly, extensively investigated area of research nowadays. We will hope that we will see those results in the near future. Thank you.
Thank you so much for the answer. In that case, please give another applause. And thank you so much for your help.
Thank you for attention.